Thank you. Thanks very much for the opportunity to be part of the symposium, which is incredibly interesting so far. I'm going to be talking about microstructural brain plasticity, and as Sonia said in the introduction, the focus of my talk is going to be on white matter. If I can get the slides to advance. There we go. So we've heard a lot about grey matter plasticity. Obviously, plasticity at the synapse is the sort of prototype of plasticity that we tend to think about when we think about learning. We think about LTP-like mechanisms of plasticity that occur at the synapse. But there's also increasing interest in the possibility that the white matter fibers themselves might undergo experience-dependent plasticity. And that could be an effective way of changing brain structure as a means to alter behavior, because if we think about the physical characteristics of white matter bundles, features such as the quality of the membrane or the quality of the myelin insulation around the axons, the diameter of the axon and the packing density of multiple axons within a fiber bundle, these physical characteristics will have consequences for the physiology of the axon bundle. Is this slide advance not working? Okay, there is a delay. So as we alter those physical characteristics, we would alter things like the speed of transmission, the quality of transmission along an axon, or even the synchronization of signals across a distributed neural network, and that in turn would have consequences for behavior. Do I have to ask you to advance the slide? There we go, okay. So we can see then that altering the physical properties of the white matter would have consequences for behavior. Sorry, the mouse really isn't responding. Yeah, I'm clicking the mouse. Okay. Should I ask you when I want to advance the slide? Okay. Okay, sorry about that, guys. So the first occasion on which we observed this was in the context of human imaging, where a number of years now ago we used diffusion imaging to look at the consequences of novel visuomotor training on brain structure. And what we found was that when naive adults learned a new visuomotor skill, in this case learning to juggle, if we compared the FA maps taken before and after that juggling experience, we could find evidence for an increase in FA, as you see by the blue clusters there, in the white matter. We also found changes in the grey matter volume in areas of the medial occipital and parietal cortices that you see there in red, and similar changes, albeit in different regions, had been reported previously. But the novel finding here was the change in white matter structure in those blue areas in underlying white matter fibers. So that was interesting because it suggested that something about the physical properties of the white matter was changing in association with that experience. But it was also frustrating, if we could advance, because we didn't know what the underlying biological characteristics of those changes are. Next slide, please. And that's because there's not a one-to-one relationship between this diffusion metric of FA and the underlying tissue properties. So this schematics illustrates just a number of possible cellular level scenarios that could underlie an increase in FA. So if we were to observe an increase in FA, that could reflect an increase in the density of the axons, a decrease in the diameter of the axons, greater coherence in the sort of geometrical organization of the axons, or could reflect some remodeling of the myelin sheath. So, for example, it could reflect formation of new myelin on previously unmyelinated axons, or thickening of myelin on previously myelinated axons. And next slide, please. If the particular interest that we have had is these properties of potential for myelin plasticity. And that's because there's been an awful lot of interest over the last 10 to 15 years or so from myelin biologists in the possibility of experience-dependent myelin change, and in fact now a very large body of evidence, much of it from in vitro preparations, which shows how alterations in the electrical properties of an axon can give rise to changes in the myelin sheath around it. Next slide, please. So this just illustrates some of the ways in which the myelin might change. So there's this sort of catch-all term of myelin plasticity captures many different potential underlying scenarios, just some of which are illustrated here. I won't go into these in detail, but the kinds of things that could happen is proliferation of new oligodendrocyte precursors, so the cells that will go on to make myelin, or mature differentiated oligodendrocytes can change the amount of myelin that they produce. And in all cases, the feeling is that activity or electrical signals along the axons will modulate the activity or proliferation of the oligodendrocytes, which will then change the insulation of the axons, thereby altering their function and potentially altering behavior. Next slide, please. So in fact, although the session is on human plasticity, the rest of my talk is going to focus on rodents, because of this difficulty of interpreting the biological basis of the human non-invasive MR signals. We've begun in recent years to perform parallel studies in rodents in which we can look not only at the MR signals but also at underlying histology to try to make better sense of what the MR signals reflect. And much of this work has been driven by Cassandra Sampar-Baptista, originally when she was a PhD student and now a postdoc in my lab. If you could advance, please. And the first study that she did during her PhD, next slide, please, was to use a skilled reaching paradigm in rats, and you can see the paradigm there in the video. It's a very, very well-characterized behavioral paradigm in which the animals learn to use a single paw to reach for a pellet of food. And you get a very nice behavioral curve over about 10 days of training in which the animals improve their skill on this task. After training on the task, Cassandra scanned the brain's post-mortem using diffusion imaging. Next slide, please. Next two. And what we found was that there was an increase in FA in the red cluster that you see here, which is the white matter underlying the sensorimotor cortex in the contralateral hemisphere to the trained paw. And next slide. And again. The rationale for doing the study in rodents is that we could then go on and perform histology that we wouldn't be able to do in humans, in this case staining for myelin basic protein, and looking in that same region of the white matter. Next slide, please. We could find increase in the myelin stain in that region, which showed the increase in FA. Next slide, please. Okay, next slide, please. We've then gone on to study that same paradigm, the same behavioral paradigm, this time longitudinally using in vivo MRI. And in this experiment, we also used diffusion imaging, but in addition, we added other imaging protocols, in particular MTR, or magnetization transfer ratio, which again isn't a perfect measure of myelin, but should provide greater sensitivity to myelin compared to FA. And what we found was a much weaker effect in FA, but interestingly, in that same area, or a similar area of white matter, we found increased MTR in the trained animals compared to the control animals, next slide, in a way that correlated with behavior. In this study, being in vivo, we could also look at functional signals, as well as looking at MT in the grey matter. Next slide, please. And looking at MT in the grey matter, we found increased MT in the secondary motor areas that you see here, and also a trend for increased MT in the cerebellar cortex, so potentially suggesting that intracortical myelin might also change as a result of this skill training. Next slide. Looking at the resting fMRI signals to quantify functional connectivity, here with a seed shown in green placed over the motor cortex, we found that the skilled animals had increased functional connectivity with the cerebellum in a cluster that you see here in blue. And next slide, that within the skilled group, there was a correlation between behavioral performance improvement and increase in functional connectivity with a range of areas, including, interestingly, the hippocampus, which relates to some of the results that Valeria was reporting, as well as the areas of the cerebellum again. Next slide. So we've then gone on to sort of ask a lower level biological question. So it's also of interest to know what the molecular correlates of these apparent microstructural changes in white matter are. So I'll assume it'll come back on again in a minute. In the meantime, I'll tell you about the study. So in collaboration with colleagues in Utrecht who carried out gene expression analysis of these same areas of white matter, we carried out a study again using post-mortem diffusion imaging where we exposed rats to a novel somatosensory experience. So if we advance the slide, you can see the behavioral paradigm here, which is, next slide please, a texture discrimination task. So the animals use their whiskers to experience a texture, and you can see the behavioral results at the bottom here. So over about 2 weeks of training, the animals improve their discrimination of the textures that they're shown. Next slide, please. So we took one of the hemispheres to scan post-mortem using diffusion imaging. Again, found increases in FA here in the yellow areas that you see, so quite diffuse regions of white matter underlying the sensorimotor system. But the other hemisphere was taken fresh to look at mRNA expression, to look at gene expression for a number of different candidate genes related to myelination. Next slide, please. And you can see, next slide, that we find an increase in the trained group in the expression of myelin basic protein. So this is interesting because it tells us more about the basic building blocks of these processes, and also potentially identifies molecular targets that could be used for interventions to modulate this process of activity-dependent myelination. Next slide, please. So finally, everything that I've shown you before so far, whether it's imaging or histology or mRNA expression, is sort of observation of the things that change in the brain in association with experience or learning. So none of those results tell us whether the changes that we're seeing are actually causally necessary for the learning or the behavior to take place. So to get that level of causal evidence, we need to interfere with the system in some way to see whether interfering with the candidate process impairs the animal's ability to learn. So we've been interested then in trying to interfere with this process of activity-dependent myelination. And to do this, we've collaborated with Bill Richardson at UCL in London, where Bill and colleagues had characterized a very interesting conditional transgenic mouse in the paper that you see here, where they, in this conditional knockout, they allow the animals to undergo normal developmental myelination, and then at the end of development, they induce a tamoxifen-induced knockout targeting the myelin regulatory factor gene, which impairs the animal's ability to make new myelin in adulthood. So they have normal developmental myelination of the motor system, but they can't differentiate new oligodendrocytes following this manipulation. And what Bill showed very interestingly in the original paper was some very nice histological evidence validating this model, and also some intriguing behavioral evidence, next slide please, which showed that the animals were impaired on this complex wheel running task that you see here. So it's like a regular running wheel, but with certain rungs removed. And so the animals, as you can see from the behavioral data on the top left there, improve their running speed over time. And what he found was that the knockout shown in red was slower on this task. And he proposed that this suggested that they had a motor learning deficit. So we've been interested in studying these mice, and we've had them in the lab for a few years now. Next slide, please. And my student, Malte Kalla, has tested a number of different cohorts now to try to get a better handle on this behavioral phenotype. And what he's now shown very convincingly, as you see from the behavioral data here, is that there is indeed a group-by-time interaction, which was the key fingerprint that we were looking for to characterize this as a learning deficit, and also that on a number of very low-level motor control tasks, there's no impairment in these animals. So, to summarize, experience alters white matter structure in the human brain, but relating those imaging changes to underlying biological events is very challenging. And we found that studies with imaging and histology, which are possible in rodent models, suggest indeed that activity-dependent changes in myelin might play a role in learning. And finally, next slide, I'd like to acknowledge my colleagues, next slide please, who did the work, so all of the colleagues in my group, particularly Cassandra Sampao-Baptista, who was involved in all of the rodent studies that I presented. Thank you to our collaborators and funders, and thank you for your attention.